In 2007, a researcher at the University of California, Los Angeles, named Naomi Eisenberger, put volunteers into a brain scanner and had them play a simple ball-tossing game with two other players. After a few rounds, the two other players stopped throwing the ball to the subject. They just stopped, excluded them from the game without explanation. The brain scanner recorded what happened next. The region that lit up was the dorsal anterior cingulate cortex, the same region that processes the sensation of physical pain. Not metaphorically similar. Identical tissue. Identical activation pattern. The anterior insula, the region that registers the distressing, urgent quality of pain, the part of your brain that makes pain feel like something you cannot ignore, activated simultaneously. Eisenberger published the findings in Science. The paper's title asked the question plainly, does rejection hurt? The scanner had answered in millimeters of activated cortex. This is not a metaphor. It is a measurement. And Eisenberger has since pushed that finding further in ways that make the overlap even harder to dismiss. In a separate experiment, she gave volunteers either acetaminophen, the active compound in Tylenol, or a placebo and had them take it daily for 3 weeks. The group taking the painkiller reported significantly lower levels of social pain on daily diaries. Then she scanned them again during a social exclusion task. The acetaminophen group showed reduced activation in the dorsal anterior cingulate cortex compared to placebo. A drug designed to block the molecular machinery of physical pain reduced the neural response to rejection. The brain is not using social pain as a metaphor for physical pain. It is using the same hardware. What science has spent the last 3 decades methodically establishing is that loneliness, not depression, not sadness, not grief, but the specific subjective experience of feeling socially disconnected, rewires the human body at a molecular level. It changes which genes are expressed in your immune cells. It alters your cortisol release patterns in ways that take years to reverse. It suppresses the class of immune cells that hunt down cancer and viral infections, while simultaneously amplifying the inflammatory response associated with heart disease, Alzheimer's disease, and type 2 diabetes. These are not psychological side effects. They are primary physiological events. The feeling and the biochemistry are happening simultaneously, triggered by the same neural signal. You cannot have one without the other. The distinction that most people draw between emotional suffering and physical suffering is, at the cellular level, not a distinction the body recognizes. Before going further, one distinction matters. Being alone and being lonely are not the same thing. Solitude, chosen, comfortable, restorative, does not produce the biological signatures described here. Monks, writers, and researchers who spend long periods working in isolation without a subjective sense of disconnection do not show the same physiological patterns. The variable the body is tracking is not physical proximity or its absence. It is the felt sense of social safety versus social threat. This distinction is not semantic. It is the entire mechanism. What makes this harder to accept is that we live in a culture that treats loneliness as a subjective complaint, something a person either manages or does not manage, a weakness dressed in the language of mental health. The science says the body does not process it as optional. John Cacioppo spent 30 years studying loneliness at the University of Chicago. When he began in the early 1980s, the field barely existed as a serious research program. Loneliness was considered an emotional state, potentially interesting to psychologists but not biologists, not immunologists, not cardiologists. It lived in the softer margins of the research world, and Cacioppo's early grant applications were reviewed with skepticism by committees whose expertise was in concrete physiology. He persisted anyway, with a systematic rigor that eventually became impossible to argue with. He developed measurement tools. He ran longitudinal cohorts. He collaborated across disciplines at a scale that changed what questions those disciplines asked. By the time he died in 2018, he had generated enough precisely measured, independently replicated data to fundamentally reframe the question of what loneliness actually is. One of his most durable findings concerned cortisol. Lonely people have elevated evening cortisol levels compared to non-lonely people. Not elevated morning cortisol, the pattern is inverted, and the inversion is significant. Your HPA axis, the hypothalamic pituitary adrenal circuit that governs cortisol release, runs on a 24-hour rhythm that in healthy individuals produces high cortisol in the morning to mobilize glucose, sharpen attention, and prepare the body for the demands of the day, then tapers to near zero by late evening. That evening low is not passive rest. It is a biological window. The cellular repair processes that rebuild damaged DNA, consolidate immune memory, and regulate synaptic density in the brain require the hormonal quiet of low cortisol to operate. Cacioppo's team found that in chronically lonely individuals, evening cortisol remained significantly elevated, measured across multiple cohorts, including the Chicago Health and Aging Project, which tracked more than 800 older adults over several years. The body is spending its repair window maintaining vigilance. Growth hormone secretion, which normally peaks in the first deep sleep cycle and drives tissue repair, is suppressed when cortisol is high. The maintenance work gets deferred. Deferred night after night, over months and years, the cumulative cellular debt is measurable. Cacioppo's group added a further layer to this picture by measuring sleep architecture directly, not just total sleep time, but the quality of sleep at the level of EEG wave patterns. They found that lonely individuals spent less time in slow-wave sleep, the deepest stage of non-REM sleep characterized by synchronized low-frequency brainwaves, even when total hours in bed were identical to those of socially connected controls. Slow-wave sleep is the physiological window in which growth hormone is secreted at its highest daily rate, in which the glymphatic system, the brain's waste clearance network, runs at full capacity, and in which immune memory consolidation occurs. Loneliness does not just make it harder to fall asleep, though that effect is also documented. It degrades the architecture of sleep from within, compressing the stages that matter most for long-term biological maintenance. But cortisol is only the first mechanism. There is a deeper layer operating simultaneously, one that required entirely different technology to see. In 2007, Steve Cole at the UCLA School of Medicine took a different approach. Rather than measuring hormones, he measured gene expression. Specifically, which genes inside immune cells were being activated or silenced in lonely people compared to socially connected people. He used genome-wide transcriptional profiling, a technique that allows you to see, simultaneously, the activity level of roughly 22,000 human protein-coding genes across a blood sample. The scale of resolution is extraordinary. You are not looking for one or two candidate genes. You are watching the entire genome at once. What Cole found became one of the most cited papers in the field of social genomics. He called the pattern the conserved transcriptional response to adversity, abbreviated CTRA. The CTRA pattern had two distinct signatures, running in opposite directions at the same time. First, genes that encode pro-inflammatory proteins, interleukin-6, interleukin-1 beta, tumor necrosis factor alpha, were significantly upregulated in lonely individuals. These are the molecular messengers that, under normal circumstances, coordinate the inflammatory response to injury and bacterial infection. They call immune cells to a site of damage, raise local tissue temperature to slow bacterial replication, and initiate the repair cascade. In lonely people, they were running at chronically elevated levels without any injury to respond to. Inflammation without a target. The transcription factor driving most of this upregulation was NF-κB, nuclear factor kappa light chain enhancer of activated B cells, which acts as a master switch for the expression of dozens of inflammatory genes. In circulating leukocytes from lonely individuals, NF-κB was significantly more active. Second, and Cole found this the more revealing signature. genes encoding the proteins that coordinate antiviral immune response, specifically the type 1 interferon pathway and the genes that support antibody production, were downregulated, suppressed. The immune system of lonely people was simultaneously overreacting to bacterial threats and underreacting to viral ones. Two directions, one signal. Included in the suppressed category were genes governing the activity of natural killer cells, NK cells, a specialized class of lymphocyte that patrols the body continuously, scanning the surface proteins of every cell it encounters. When an NK cell detects a cell displaying the abnormal surface signatures of viral infection or malignant transformation, it kills it on contact, secreting perforin and granzyme proteins that puncture the target cell membrane and trigger programmed cell death. NK cell surveillance is among the body's primary defenses against both early-stage tumor formation and viral spread. In lonely individuals, the genes that drive NK cell activation, proliferation, and cytotoxic function were measurably suppressed. The sentinels were being stood down. Cole spent years working out why this pattern existed at all. The answer, when it became clear, was that the pattern made complete evolutionary sense. Not for modern urban life, but for the conditions under which human immune architecture was selected. When you are separated from your group, you face an asymmetric threat profile. You are more likely to sustain a physical wound from a predator, from falling, from the hazards of unsupported survival. A wound introduces bacteria. You are less likely to encounter other humans, which is the primary transmission vector for viruses that spread through proximity and shared respiration. A body optimally tuned for isolated survival should upregulate antibacterial inflammation and downregulate antiviral defense. That is exactly what happens. The immune system is executing a rational ancestral program with perfect fidelity, 300,000 years after the conditions that program was designed for disappeared. It received the signal, isolated. It ran the code. The signal triggering the CTRA pattern is not actual isolation. This is the most disorienting finding in the field. Cole and Cacioppo demonstrated across multiple independent studies that the gene expression changes are not driven by the objective number of social contacts a person has, or by the number of hours they spend physically alone. They are driven by perceived isolation, the subjective internal experience of feeling disconnected, even in the presence of other people. A person at a crowded party who feels that no one there truly knows them will show the same CTRA signature as someone with no social contacts at all. The molecular machinery is responding to the brain's assessment of social safety, not to the actual proximity of other humans. This has a consequence that took the research community several years to fully absorb. Proximity alone does not reverse the pattern. You can surround a lonely person with people and the gene expression changes persist if the internal perception of disconnection persists. Cole has stated this plainly. The intervention has to change the signal, not just the stimulus. The neural mechanism behind the signal runs through a circuit Cacioppo named the hypervigilance pathway. Lonely individuals show measurably enhanced neural responses to social threat cues, a mildly disapproving facial expression, an ambiguous tone of voice, a conversational pause that could signal rejection. Using high-density electroencephalography that measures event-related brain potentials with millisecond resolution, Cacioppo's group showed that the lonely brain processes potential social threats faster and with greater neural amplitude, while showing reduced neural activation in response to positive social cues. The nervous system has been recalibrated, and the recalibration is self-reinforcing in a way that closes the loop biologically. Heightened threat perception makes genuine connection more difficult because neutral signals read as threatening. Deepened perceived isolation sustains the CTRA pattern. The elevated evening cortisol degrades slow-wave sleep. Degraded slow-wave sleep impairs prefrontal regulation of the amygdala and the threat detection machinery. The next day's social interactions are processed by a brain that is both sleep-deprived and running heightened threat sensitivity. The loop does not break itself. The downstream consequences of running this loop for years have been measured with precision. In 2015, Julianne Holt-Lunstad at Brigham Young University conducted a meta-analysis of 148 longitudinal studies covering more than 300,000 participants, followed over an average of 7.5 years, examining whether social isolation predicted mortality after controlling for other known risk factors. The result was a 26% increase in mortality risk for individuals with inadequate social connections. The effect size was larger than smoking 15 cigarettes a day, larger than obesity, comparable in magnitude to alcoholism. These were not self-reported health outcomes. These were death records. The specific cause pathways are multiple. Chronic elevated IL-6 and TNF-alpha damage the endothelial lining of blood vessels, the single-cell thick layer that lines every artery in your body, and accelerate the formation of atherosclerotic plaques over years. Framingham Heart Study cohort analyses found significantly elevated rates of cardiovascular events in socially isolated individuals after controlling for traditional cardiovascular risk factors, including hypertension, cholesterol, smoking, and physical activity. The suppressed antiviral response has been directly tested. Sheldon Cohen at Carnegie Mellon University exposed volunteers to a rhinovirus through nasal drops after precisely measuring their social network characteristics. Participants with fewer types of social relationships were 4.2 times more likely to develop illness at the same viral dose. The virus was identical. The immune preparation was not. The connection to neurodegeneration is more recent, but mechanistically coherent. Researchers at the Rush Alzheimer's Disease Center found that lonely individuals had a 64% higher risk of developing dementia than those with satisfying social connections after controlling for objective social network size. The mechanistic chain runs through neuroinflammation. Chronically elevated IL-6 crosses the blood-brain barrier and activates microglia, the brain's resident immune cells, in ways that degrade synaptic pruning accuracy and may accelerate tau protein aggregation. Whether this constitutes causation or a shared upstream mechanism is an active research question. The biological plausibility is not in question. Now I want to bring you back to the brain scanner at UCLA. The participant is watching the ball passing between the other two players, not to them. The dorsal anterior cingulate cortex activates. The anterior insula activates. These regions were not recruited by evolution to process social slights. They evolved to register tissue damage severe enough to require the organism's full attention. The class of pain that, if you ignore it, kills you. The brain drafted them into service for social disconnection because, in the conditions under which human neural architecture was shaped, social disconnection was tissue damage. It was slow death from predation, from exposure, from loss of the cooperative food sharing and collective defense that made human survival possible at all. The neural alarm proportionate to a broken bone is proportionate to what being genuinely cast out of a group actually meant, across most of the time our species has existed. Steve Cole has said that what struck him most about the CTRA data was not how dramatic the changes were, but how conserved the pattern was across every population he examined. He found the same gene expression signature in HIV patients experiencing social stigma, in cancer patients treated in isolation wards, in people living in high-crime neighborhoods who did not trust their neighbors, and in bereaved individuals in the months following the death of a spouse. The body runs one ancient molecular program in response to the perception of isolation. Amplify inflammation, suppress viral defense, raise evening cortisol. It has been running that program for longer than our species has had language to describe the feeling that accompanies it. The reversal requires time. Cole's lab found that even after sustained interventions, cognitive behavioral therapy targeting hypervigilance, structured community integration programs, and group activities designed to build genuine rather than superficial social bonds. Gene expression profiles required months to years to fully normalize. The inflammatory genes are not easily convinced. The nervous system accepts the signal that the threat has passed slowly, the way bone rebuilds density after injury, through accumulated evidence, over time, without shortcuts. You felt something shift the last time you sat in a room full of people and felt entirely unknown. That was not only a feeling. Your cortisol was rising at a time when it should have been falling. Your NF-κB transcription factor was binding to the promoter regions of inflammatory genes and calling for the production of interleukin-6. Your natural killer cells, the sentinels that scan your tissues for tumors and viral infection, were standing down. The ancient molecular program was reading one signal and running its one response. Isolated. Prepare for a wound. The feeling and the chemistry are the same event. There is no version of loneliness that exists only in the mind. There never was.